Adaptive immunity includes humoral and cytotoxic immunity. And these two forms of immunity use antibodies, immunoglobulins, and direct attacking cells um, to neutralize a pathogen. So, cytotoxic immunity focuses on making an army of T cells that can kill the actual infected cells. Unlike humoral immunity, which relies on B cells to produce immunoglobulins that will then mark the pathogen for destruction, cytotoxic immunity uses the T cells to recognize and directly destroy the pathogens. T cells come in about 15 different types, I think, probably more at this point. And each of those T cells has a very distinct role. So, cytotoxic T cells, which are also called CD8 T cells, they attack and directly destroy infected cells, but their actions are regulated. So, regulatory T cells, sometimes they used to be called suppressor T cells, those will stop the cytotoxic T cell activity when necessary, kind of preventing like an overreaction, which are usually included in a suite of diseases called autoimmune diseases. Additionally, there are also memory T cells, which are created, and they recognize the antigen for future encounters. So, what they do is they kind of enable a faster immune response of the cytotoxic T cells. All T cells have what are called T cell receptors, which identify them as T cells. Naive T cells, which haven't yet encountered their target, they mature into CD4, which are called helper cells, and CD8, which are called cytotoxic T cells. All T cells mature in the thymus, which I have a picture of here. They undergo positive and negative selection. And what that does is it ensures that they can recognize foreign antigens for destruction and that they also don't recognize self antigens for destruction. The first step in mobilizing the cellular T cell army, if infection is present, it is you have to present the antigen to naive T cells that are just kind of like hanging out in the lymph nodes. An APC, an antigen presenting cell, here I have a dendritic cell, drinks foreign antigen by pinocytosis. And then the dendritic cell moves into a lymph node, and you can see it accessing the lymph node by the afferent vessel here. In the cortex of the lymph node, the APC displays the epitope of the pathogen on its class 2, on its MHC class 2 protein. And this is the dendritic cell or the antigen presenting cell saying, "Hey, I found this." Okay, so I mean, we've presented an antigen. What we want to do is now make a whole bunch of T cells that are like primed to destroy it. But before we do that, we have to have this co-stimulation. And this is the process of the T cell binding to a second receptor protein in the cell membrane of the APC. This co-stimulation protein, it ensures that we're not going to like induce an immune response when there's no pathogen present. And again, in fact, these co-stimulation proteins play a big part in autoimmune responses where an immune response is elicited but no pathogen is present. So basically, co-stimulation is just the T cell making like 100% sure that it should go ahead and clone itself. Um, there are essentially three signals that have to happen for a T cell to become activated and clone itself. Signal one is the binding of the T cell receptor to the MHC class 2 protein on the APC. Signal two is the binding of a T cell co-receptor to another receptor on the APC cell surface. The binding of cytokines to their receptors on the T cell represent signal number 3. So the APC will secrete cytokines that further activates the naive T cell. Different combinations of these cytokines can influence T cells to differentiate into very specific effector T cell subtypes that produce very specific cytokines. Once the T cell makes sure that it should go ahead and clone itself, it does. And it makes an enormous army. And among the cells in the army are cytotoxic T cells that will just go forth and destroy. Regulatory and memory T cells are also made. So cytotoxic T cells in a lymph node that are activated will leave the lymph node, enter the blood circulation, and then it's kind of like drawn to infection areas by other cytokines and interferons. The cytotoxic T cell, when it binds to the foreign cell, it kind of like pokes holes in it, and it injects enzymes and toxins to break up the cell membrane and like just explode the cell. Cytotoxic T cells use proteins like granzymes and perforin. Perforin creates pores in the cell membrane of the infected cell, and it just leads to, as I said, their complete destruction. In viral infections, perforin causes the infected host cell to burst. And just as dying virally infected cells, cytotoxic T cells will release interferons. And remember, interferons were kind of like that signal, like the ship is going down, I've been infected, beware. There's also TNF, or tumor necrosis factor, which activates macrophages, and that is released especially when cancer cells are encountered. And additionally, as if all of these things weren't enough to destroy a cell, additionally, cytokines can induce apoptosis or programmed signal death. And it can induce that in the infected cell or in nearby infected cells. There's also lymphotoxins, which is another tool that T cells can use. And lymphotoxins, they have to get into the cell, and then they disrupt ATP production. And this incapacitates the infected cell. In the end, the immune response relies heavily on CD8 T cells to do the actual killing. CD4 cells, or helper T cells, they help or facilitate recognition of infections, while CD8 cells are responsible for the direct killing of the infected cell or pathogen. So meanwhile, while CD8 cells are working, CD4 cells are more concentrating on coordinating communication between various immune cells, not just helper T cells, but basophils, macrophages, neutrophils. You can see 4 different types of CD4 cells in this picture here. You can also see that many of them secrete specific interleukins that then activate other cells. It also secretes an interferon, which can activate macrophages. So helper T cells, what they do is they go up to APCs, like macrophages, and they say, hey, we're looking for this. If you find it, let me know. And in the process of speaking with just one APC, the helper T cell will release cytokines, and that draws close any NK cells, leukocytes. Those cytokines can start inflammation, and they can invoke any other innate defenses that could be helpful, like fever. So helper T cells, they can also tell B cells to produce antibodies, which then mark pathogens for invasion and make that pathogen easier for the CD8 T cells to find and destroy them. Cytokines also act as distress signals, right? Calling for help to eliminate invaders, especially in areas where they're concentrated. When a helper T cell encounters an APC with an antigen, the helper T cell will release interleukins, and that's that IFN gamma factor there. And these interleukins do three specific things. They attract neutrophils and NK cells to an area. They attract macrophiles and sequester them in the area. And they also stimulate B and T cell mitosis. Every time we use adaptive immunity, we generate memory cells, and that's part of it being adaptive. Both CD8 T cells, cytotoxic T cells, and CD4 helper T cells, they contribute to the immune response by creating these memory cells and then increasing their numbers. These memory cells are longer lived. They can activate more quickly, so it takes fewer steps for them to produce their army of cells. And this ensures that the second time that we encounter the same pathogen, the immune system responds quickly and effectively. It's as if the immune system says, I know who you are, and I know what cells to activate to eliminate you. And this more rapid response is called a T cell recall response or a secondary immune response. This is the idea on which vaccines are predicated, that with the vaccine you do not receive the pathogen itself, but you receive pieces of it. You develop a primary immune response, and then the second time you're exposed to the pathogen, you don't even know it because you produce a secondary immune response. Now, that's passive immunity when you receive a vaccine of an attenuated virus that's all chopped up. There's also active immunity, which is like when you have a chickenpox party and you bring your child over to meet with another child who is infected. You evoke a primary immune response, and then the next time your child's exposed to chickenpox, they have a secondary immune response and they don't even realize it.